Welcome to AP Biology, Topic 7.2, Natural Selection. I'm Gail Fizer. I'm from Darnell Cookman Middle High School in Jacksonville, Florida. A message from the College Board. Today, the topic we are covering is one that colleges have asked that all AP students study, even if it will not be on the exam. Colleges are supporting AP students this year by allowing this more flexible approach to the exam, where it will focus on a lesser amount of content than in other years. But in turn, they have asked that AP students learn today's material. To make this relevant to your upcoming exam, we'll also include some exam prep in today's lesson. In a week or so, after we've covered some of these key outstanding topics, we'll be posting the full schedule of review topics for the remainder of this course so that you can choose which topics you want to prioritize for review and thus which classes you'll want to attend. So first, I'm going to go over a lesson overview. So today, I will be introducing you to the principles of natural selection, focusing on phenotypical variation. And then we'll also be going over science skill 1B, which has to do with explanation. You'll be gaining proficiency in explaining biological concepts and processes. You should already be familiar with the causes of natural selection, how natural selection affects populations, explaining biological concepts and processes. By the end of this lesson, you will also be able to describe the importance of phenotypic variation in a population and explain biological phenomena. So we'll have our warm-up first. So this is our homework from our last lesson, which was 7.1. And so we're going to start off every lesson with a warm-up, and it will be from the homework from the previous lesson. So this will be from 7.1. And before I will give you time after I go over it for those that are just joining us for the first time tonight. I'll go ahead and tell you my strategy of going through questions, whether it be multiple choice or FRQ. I take an approach of looking at the diagram, and then I want an interpretation of that diagram to tell me the story of what's happening before I go deep into the question. So an example of me doing that and teaching my students to do this would be I would start with, I see 3 boxes. I see that we have blood, epithelial layer, intestinal lumen. I see there's a cell in the epithelial layer, and then there on the right is the intestinal lumen. So I must be in the, this must be a cell in the intestine, and then on the other side has to do with the absorption into the blood. So now that I know where I am and I can see villi on the cell, now I can look at the sodium. I see high sodium in the lumen, and then low in the sodium. That's already ringing a bell with me that we're going to be moving sodium from a high concentration to a low concentration. And then I see sodium high in the blood. So we'll be going from a low sodium to a high sodium from the epithelial layer to the blood. And that's going to tell me that we're going against the gradient and that I'm going to have to use some ATP to make that happen. Then I see the glucose. There's a medium amount of glucose in the lumen, there's high in the epithelial layer, and there's low in the blood. And so I know that the high glucose to low glucose, that will move in a concentration gradient, but somehow I have to get this medium glucose in the lumen to that cell. So now I'm looking at the diagram, and I see that sodium is moving from a high concentration to a low concentration through a transport protein. And while it's doing that, glucose is moving also. So it's acting as a co-transport into the cell. And now once we're in the cell, the glucose is now at a high glucose amount, and it's going to move out of the cell from high to low concentration, still through a transport protein. Now, the sodium on the inside, I said earlier, that was low sodium, so we're going to have to use the sodium-potassium pump and use some ATP to go against that gradient. So now that I kind of know the story and what's going on, now I'm going to see what the question is going to ask me. Generally, I would read the question, and then if it was a long passage, that I would go into the into the text last, the passage last, but this is a very short one, so I'm just going to do it all at one time. Okay, it says glucose and sodium move from the lumen of the small intestine into the blood via transport proteins in the epithelial cells lining the small intestine. Based on figure 1, describe the direct source of energy used to move glucose into the epithelial cell from the intestinal lumen. And now there's another, that was our first task word, that was describe. We went over describe in the previous lesson a lot. And then we also covered explain in just a small amount. So explain how this system maximizes glucose absorption from the intestinal lumen into the epithelial cells and from, down there, that and is key because not only we're explaining the first half of that sentence, we're going to need to explain the second part too, and from the epithelial cells in the blood. And there's also another key word in there, explain how this system, so how this glucose is working from the lumen to the cell and back and out to the blood all together. So I'm going to give you a minute and I'm going to time you, and you go ahead and start answering that question. On every warm up, you're going to need a pen and a paper available. So, because I'm going to I'll wait on you for a minute and then I'll tell you when I'm going to count down to 10 seconds and see how that works today. So go ahead and give that FRQ question a try. Okay, I'm going to give you 10 more seconds. Okay. Now, I know that wasn't time enough to answer this question. And if what I will suggest is that after this live viewing of this video, that you watch it in pieces and slow it down at your own pace and redo everything that we're doing. And so you can work through them at a more comfortable pace. So check your answers against the scoring guideline. There are 3 points here. There's a describe point, which you should have addressed that the energy from the sodium gradient is what is the source of energy to move the glucose into the cell, okay? And then there's 2 explains, remember that and, so there were 2 places to explain. So there's 2 points here, and you should have addressed that the sodium-potassium pump maintains a sodium concentration gradient and allows for co-transport of the glucose. And then you should have also addressed that the symport, or the inflow of glucose, maintains a glucose concentration gradient between the epithelial cells and the blood and allows for facilitated diffusion of the glucose. All right, I hope you did a good job. All right, so I'm going to do this for the first few videos that we have. So as people start to jump on and be regular, then I won't tell you this every single time. But this is how the framework of the course. And so you've been working through different units this year: biomolecules, cells, photosynthesis, and we have broken those units into small topics. And each topic has questions called the learning objectives. And this is what you need to be able to do. And we need you to know content in order to be able to do that. So the content is called the essential knowledge, and each topic has an associated science skill that will be tested along with it. So this is how we're going to show we know the answer to the question. So the science skill that is associated with natural selection is visual suggested science skill. For this lesson is visual representation. And you've probably been doing this in class all year anyway. So today we'll topic skill pair, we'll do natural selection and explain together. So what you need to know. Okay, this is my favorite part, is I had to stop putting in more material today because there's just so much that I enjoy that what I'm about to talk about. So as from our previous lesson, well, we learned that natural selection is a mechanism of evolution. And also now tonight, we're going to talk about natural selection, phenotypic variation, and selective pressures. And I like to teach by examples. And so I have two examples that I'm going to talk about. I'm going to talk about the peppered moth, and I'm also going to talk about sickle cell anemia. And the peppered moth is very simple. It's a very simple story. In fact, I did have a video that was connected to this, but I was afraid of music copyright laws getting me. So anyway, about the second week that my kids were separated from me and we were not in school, that I started missing them, and I was thinking I need to teach them some science, and I need to make it in this harsh and not harsh, in a very sweet and soft way that they'll enjoy and think it's fun. So what I'm about to show you is at a very elementary level, but it tells the story perfectly. I do have a video on YouTube that I did send them if you could find it. Anyway, so we're going to talk about the peppered moth. So what we have here, we have moths that have two phenotypes. We have a peppered and a dark moth. And a phenotype, remember, is the observable physical trait, okay, or characteristic, if you want to call it that. Now, their environment of the story that I'm about to tell you is on a light-colored birch tree with lichen. So, can you see them? This peppered moth population has adaptations selected for in this environment. If you can see them, there are 6 of them that I placed on there. There were a higher number of peppered moths than dark moths. Birds are able to see the dark moths on light-colored trees. The dark moth has a phenotype selected against in this environment. And there is a lot of research about what I'm about to tell you. And this is the birch tree before the Industrial Revolution. And this is the birch tree turned dark from burning coal during the Industrial Revolution. Now, the peppered moths were detectable by the birds on this dark birch tree covered in coal. The birds now began eating the peppered moths. And now there were more dark moths reproducing than peppered. The dark moth phenotype was selected for in this environment. The peppered moth was selected against the dark, wait, hold on just a second. Sorry about that. The dark moth phenotype was selected for in this environment. The peppered moth was selected against. I didn't see my period. All right, the dark moth populations had adaptations that increased their survival and reproduction. Now, the peppered moths now had a lower population than the dark. And then that's when efforts to clean the air began. Now, the lighter environment allowed the birds to see the dark moths again. The dark moths were selected against in this environment again. And the takeaways from this example: natural selection acts on phenotypic variations in populations. Environments can change and apply new selective pressures to populations. Some phenotypic variations significantly increase or decrease fitness. This is reproductive success of the organism in particular environments. So I sent this is really sweet and cute in this fast little video that I made for my students on this topic. All right, so now let's get to sickle cell anemia, and there is so much science that I love in this topic. First, we have 2 different phenotypes of red blood cell here. We have a normal red blood cell, and we have a sickled red blood cell. And if you look in this blood smear over to the left, you can see that there are sickled cells that you could pick out, okay? And this is what happens, is that what a patient feels like. I used to be a nurse a long time ago before I was a teacher. Anyway, so there is this sickling that's going to happen, and I'm going to tell you how that happens. But anyway, there are characteristics of the membrane that change, not only just in this shape, and I'll tell you how that shape changes, but it starts sticking to the endothelium, and they start sticking together, and they end up blocking the good red blood cells that are able to carry oxygen. And so any cells past that point, they start having a decrease in availability of oxygen, and so they are starving of oxygen, and then sometimes they actually go through infarction and they die, and then those cells are unable to function anymore, and there's a lot of pain involved in that. So this is how it all starts. Okay, so this blue strand is DNA, and that is a template strand, okay? What you're not seeing is the coding strand, because you already see the messenger RNA complementary to that template strand. So there's the DNA, and you can see that there's one that's normal hemoglobin and one that's mutated hemoglobin. Now, in the normal hemoglobin, in the coding strand, okay, you should have a GAG for a particular codon. Well, there's this one single point mutation which happens, and it ends up being a GUG. The thing is, with the GAG, it codes for glutamic acid, and the GUG codes for valine. So what should be there in a normal hemoglobin is the glutamic acid, but ends up with a valine instead. Now, a single point mutation on chromosome 11 is responsible. Thymine is substituted for an adenine. So what you're seeing is this, I don't know if you can see me move this cursor, but what you're seeing is a U, okay, when they're talking about a thymine is substituted for an adenine, but that would be on the coding strand if I had a coding strand here. Okay, ultimately responsible for producing the hemoglobin protein. This one single point mutation results in the production of the valine amino acid instead of glutamic acid. So this is called missense. Now, this is, I'm going to give you a question later that this diagram is from. So let's go through what's on this diagram. There is a ribosome, and I see a large subunit and a small subunit, and I can tell that my messenger RNA is going through my ribosome, and I can tell it's heading in a 5' to 3' direction going in, because this is a codon 2, 3, 4, and 5, okay? And this was my amino terminal in, and I have amino acid 1, 2, 3, and 4. So these are transfer RNAs, and here we have our GAG. So I'm talking about normal hemoglobin. So this right here is what's supposed to be here. This codon is for GAG, and if we look on our genetic code down at G, this is how you read it. There's a G, and there's a G, and there's a G. So there's GAG right down here, and that's going to code for glutamic acid, and that's what is supposed to be there, okay? That's normal hemoglobin. So now let's look at an example with the mutation, I'm sorry. All right, so same situation. We have a ribosome, we have transfer RNAs, we have messenger RNA going through the ribosome, and here we have our GUG, okay? So there we have that mutation that you should not be there, that should be an A, right? Okay, so now we're going to be complementary to that anticodon, which is CAC, and that anticodon is responsible for bringing the amino acid valine into the ribosome to be part of the next, or the polypeptide that it's building. So let's look at that. Here's a G, U, and a G. Now we have valine. So that's how we ended up with the wrong amino acid. All right, so amino acids have different properties, and they'll cause them to interact differently. So what happens is, if you see this long fiber, these guys are not supposed to be in a rod like this. They're supposed to be single hemoglobin units. Anyway, these mutated hemoglobin molecules now stick together and form a fiber rod, which gives the blood cell the sickled shape. So think about putting a growing rod in this red blood cell, and how it would like eventually like push the plasma membrane around, and that's how we're forming the sickled shape. Now, I talked about them having the amino acids having different properties. So let's go over the basics of this amino acid, because it's probably been around a long time since you have, if you started with biomolecules at the beginning of the year. So here we have an amino acid, and we have the amine functional group on one side, and we have the carboxyl functional group on the other. There's our central carbon, and here is our R group, is what we're going to call it. Okay. Here is our amine N, carboxyl N, here's our central carbon, and our R group, which is very different. Each of the 20 have a different R group, and they have, they're grouped into different properties. And if you look at the glutamic acid, it's hydrophilic, and the valine is hydrophobic. And so what I tell my students is, if we all stood up and we were amino acids in a protein, where would the hydrophobic members be? And they say, well, we'd be in the inside of the group, right? Because as a cell, we're surrounded by water. So the hydrophilic ones would have to be on the outside. So I said, what if we changed the hydrophilic on the outside and gave them a substitution and then made them hydrophobic? What would happen? And they say, oh, they would all move to the center. Right, so that's going to change the structure. And when you change the structure of a protein, then you're going to change the function as well. Okay, so I added a little bit about how you get sickle cell through and genetic inheritance of it. And so I drew a little simple Punnett square. And so I could say uppercase S, lowercase s, but I'm going to just do it like I usually do in my room. And we have a big S, little s. Okay, so the father is heterozygous and the mother is heterozygous. And if we have offspring from them, we can expect that 25% of them would be homozygous dominant, okay, with the normal hemoglobin. S in the uppercase is going to represent the normal hemoglobin, and the lowercase is going to represent the mutated hemoglobin. All right, so we have a 50% chance of having a heterozygous offspring, and we have a 25% of having a recessive offspring. Okay, variation in fitness. Homozygous dominant, you have 2 normal alleles. If you have 2 normal alleles, then the parasite responsible for malaria can use your red blood cells to reproduce, and then you'll have malaria, and you'll be very sick. Homozygous recessive, there's 2 abnormal alleles. If you have 2 abnormal alleles, then you will get sickle cell anemia and no malaria. The parasite cannot reproduce in the sickle cell. And then you have the heterozygous carrier. You have 1 normal allele and 1 abnormal allele. And if you have 1 normal and 1 abnormal allele, then you're a carrier and you'll have the heterozygous advantage. No malaria, no sickle cell. So there are different outcomes for different phenotypes. Homozygous recessive gets the sickle cell anemia that I just talked about, and oxygen is not delivered efficiently. Your blood cells die young and you become anemic. Tissues die, stroke and organ failure can happen. When you're homozygous dominant, then you're able to get malaria. And if you have the heterozygous advantage, then you won't get either. If you're in an area with a high incidence of malaria, being homozygous recessive or heterozygous would protect you from getting malaria. Okay, so now for the science skill explanation. Today's task verb is explain. So we're going to learn how to explain biological concepts and processes. All right, explain. Explain is how or why a process occurs. And it can also include comparing patterns and or trends in a representation, and or predicting patterns and or trends in a representation. So let's have a little check for understanding. And I want you to take a minute to tell me. I'm going to get my phone out. You can start writing now, and I'm going to time it, and I'll tell you when you have 10 seconds left. Okay. And Okay, go. I want you to explain the effect of color on the fitness of the peppered moth and the dark moth. Okay, 10 more seconds. And I know you need more time, perhaps. Okay. All right. So let's see how your answer compares. All right, some things you might have said. There's not just one right answer. You may have said environmental changes put different selective pressures on the moth populations. Dark moths have increased fitness or reproductive success. I always want you to follow up fitness with talking about what it means on dark trees because birds will not see them and eat them. Peppered moths will have increased fitness on white trees. Dark moths will have decreased fitness on dark trees. Phenotypic variation can increase or decrease an organism's reproductive success in particular environments, and natural selection acts on phenotypic variations in populations. So let's work through some problems. I have a couple of multiple choice that we're going to go through, and it's still, even though your exam is, whoops. All right. All right. So even though your exam is going to be FRQs, interpreting a multiple choice is the same thing. Okay. We don't have a diagram or table or graph in this particular question, but we still have text to interpret and find out what the story is telling us. So the bird Paris major is commonly found in man-made habitats. During the summer, these birds primarily feed on insects, and in the winter, when insects are scarce, they forage in gardens and bird feeders for seeds, nuts, and berries. Recently, climate change has led to a decrease in food available from gardens, and scientists have observed that the average beak length of the birds has increased. Which of the following best explains the directional shift in beak length in these birds? Okay, so what did I just read? I just read that in the summer they eat insects, in the winter they have to go to gardens for bird feeders for seeds, and now that's not there. So they need to find something else to eat, right? And their beak is and the average beak of the population, not just a bird, because we're not just going to say a bird has another beak, and you know why. Anyway, so which of the following best explains the directional shift in beak length in these birds? Our options are A, longer beaks allow the birds to better fend off predators. B, longer beaks increase the fitness of the birds in wild habitats. C, longer beaks allow the birds to better access seeds and bird feeders. And D, longer beaks decrease the fitness of the birds in wild habitats. I'll give you just a minute. I don't think you need a minute for this one. Okay, 10 seconds. Okay, I hope that you chose answer B. Okay, here's the second question, has to do with climate change also. All right, climate change is disrupting the length of winter in various ecosystems, resulting in less snowfall, and milder winters. Scientists in Finland have been observing the tawny owl, a fairly large owl species that is commonly found in woodlands across much of Europe and Asia. Typically, the feathers of the upper body are either brown, and I'm going to say big B, and little b, or gray, little b. Scientists study tawny owls in Finland and have found that the percentage of tawny owls that have brown feathers has increased over the past 28 years. Which of the following best explains why the brown feathered phenotype in tawny owls has increased over the past 28 years? Think about the length of the winter and less snowfall. Oh, I'm going to read you those. Okay. A, since the brown genotype is dominant, it will eventually eliminate the recessive gray genotype from the population. B, darker feather color in tawny owls makes them less visible to predators as they sit in the trees. C, with milder winters, the advantage of gray feathers then blend into a snowy environment that has decreased. And D, the directional selection toward a darker phenotype is random and likely due to a bottleneck effect. All right, take just a moment. I think we'll stop at 30 seconds on this, so 10 more seconds. Okay. I hope you chose answer C. All right, now you need to write a little bit, just a sentence or two. All right, so now Charles Darwin proposed that evolution by natural selection was the basis for the differences that he saw in similar organisms as he traveled and collected specimens in, I just need to learn how to do that. All right, as he traveled and collected specimens in South America and on the Galapagos Islands. And now you need to explain, and explain is our task verb now, okay? So remember that was how or why. The theory of evolution by natural selection as presented by Darwin. So explain the theory of evolution by natural selection, okay? I'll give you a whole minute to do that. Okay, 10 seconds. Okay. All right, let's see what the scoring guidelines said for this one. Okay, there are a number of things that you could talk about, okay? You could talk about reproductive potential, which is the ability to overproduce. You could talk about variability, inheritable changes or mutations linked to variability, limited resources, whether biotic or abiotic, competition, interspecific struggle for existence, differential reproduction, reproductive success of the variants, generations, time needed for evolution to occur, and elaboration. You could have expanded on Darwin's ideas such as effects of environmental change or artificial selection, or given a good linked example of so. Something that I want you, when you're talking about reproduction, and another good thing to always add in is about that it's not just happen, that it's multiple generations. Sometimes that's a good thing to add when you're talking about evolution and also experimental design. Okay, so I love this question. I just found it this morning. Anyway, I love it. And so I'm going to look at the diagram and I'll talk you through the diagram. And then it has 4 parts to it. And I want you to spend 20 minutes on it at home, but not now. And so anyway, we'll just run through it real quick and I'll just guide you through it, okay? All right, so my diagram, I can see that there are 3 steps to what the question wants me to answer. I see a large ribosomal unit and a small ribosomal unit. I see that I have my messenger RNA. I see that we have some transfer RNAs bringing in some amino acids. We have codons 1, 2, 3, 4, and 5, as in triplets, as in those codons are those are representing 3 nucleotides, okay? All right, so they're called a codon or a triplet. I sometimes use that. All right, so if you have number 3 if you look above it, it's at the 3rd amino acid, and there are 2 above it, which means number 1 and number 2, their amino acid has been taken care of, and it's already in the polypeptide chain. Now we have, if you remember, you have an A site and a P site and an E site. And I tell my kids it's an arrival site, a polypeptide building site, and an exit site. So in the second one, I still see them in the same location. In step 2, I see a transfer RNA bringing in, now it has just attached, what has just happened is that the amino acid that came in on codon 4 has now been added to the rest, okay? And now it is on the codon 4's transfer RNA that's complementary to that. So you see that number 3, that they have shifted. Now they're in the polypeptide place, and then they're in the elongation, and then you have them in the exit without an amino acid, because now it has become part of this building of this polypeptide. So now let me read you the story. A processed eukaryotic messenger RNA lacking introns. So pay close attention to those words that I just said. It's eukaryotic messenger RNA, and it's lacking introns because there's been a nucleus, okay, and inside that nucleus there has went from messenger RNA through some processing steps, and the introns are still were taken out, were cut out inside the nucleus before the messenger RNA, which was a process, came out and exited the nucleus. Now, the messenger RNA is introduced into the bacteria. So now we're talking about eukaryotic messenger RNA being put into bacteria so that the bacteria can produce a large quantity of the encoded polypeptide. Translation of the messenger RNA to produce a polypeptide involves the binding of the large and small ribosomal units to a messenger RNA. Each complete ribosome has 3 transfer RNA binding sites that align with the 3 sequential codons in the messenger RNA. 3 steps in the process of translation elongation, the addition of amino acids to a growing polypeptide chain, are illustrated in figure 1. And we just looked at that, so we already know what's going on in that diagram. All right, so let's look at A and look at B. And then I'm going to give you a few moments to answer those 2, and then we have a C and D, but I just want you to get a kind of a planning stage outline. You're not going to be able to write this because we can't spend 20 minutes here, but I do want you to try to spend more time at home on these FRQs that I've included. Okay, so here we have our describe. A has described why translation of a messenger RNA can start before transcription of messenger RNA is complete in prokaryotes, but not in eukaryotes. And I gave you every clue in that first part of that sentence, okay? And then B, referring to figure 1, explain the process illustrated between step 1 and step 2. So that's going to be a how, okay? And describe is going to be what are the characteristics of what that question is asking about. So I'll give you a couple minutes on that. I'll give you 2 minutes, okay, because it's 2 questions. Okay, you have 30 seconds. Okay. All right. So let's move on to C and D, and then I'll reveal all of the answers at one time. Okay, now let's look. We've seen this before, right? So we already know what this is, and that's why I gave you that image before, so you can kind of get used to it. All right, so we have a large ribosomal subunit, small messenger RNA headed through 5 to 3' way. We have codons 2, 3, 4, 5. We have a building a polypeptide chain, and we have transfer RNA, but now we have blank boxes, okay? So this is your job now. And C, fill in the boxes in the template and there's also a genetic code at the bottom, and I'm going to go ahead and put a hint word in there, a universal genetic code, okay? Fill in the boxes in the template to identify the nucleotide sequence from 3' to 5' of the transfer RNA anticodon that will recognize codon 5, okay? Codon 5 is right here, all right? And the transfer RNA anticodon that will recognize codon 510, and that's right here, of the messenger RNA template. And to identify the amino acids that will be added at these 2 positions, a codon table is provided for reference. I'm going to go ahead and give you a minute to work on that before I read the next one, okay? Okay, let's go 10 seconds. Okay. All right, let's read D. Referring to the figures provided, explain why the polypeptide produced when the transformed bacteria translate the introduced eukaryotic messenger RNA provides evidence for the common ancestry of all living organisms. Okay, so you have remember, you have eukaryotic messenger RNA and you have a bacteria ribosome. And ribosomes, remember, eukaryotic cells and prokaryotic cells have ribosomes. So give it a try for D. Okay, about 10 seconds. All right, let's take a look. I'm trying to give you your 10 seconds. Okay, let's look at the scoring guidelines. So your answer for A should resemble this. Prokaryotes do not have a nuclear envelope that separates the genome from ribosomes in the cytoplasm of the cell, whereas eukaryotes do. I tried to give you a hint about that nucleus. All right, the growing polypeptide chain attached to the transfer RNA aligned with the codon 3 of the messenger RNA is transferred to the amino terminus of amino acid 4 attached to the transfer RNA aligned with the codon 4 of the messenger RNA. So all you needed to do was write down each step of the way what exactly was happening to answer B. All right, for codon 5, the anticodon sequence is GCC, and the amino acid added is arginine. The response also indicates that codon 510, there will be no transfer RNA with a complementary anticodon because UAG in the messenger RNA is a translation stop sequence and no amino acid will be added and the translation terminates. Okay, and then D should resemble the polypeptide produced by translation of a processed eukaryotic messenger RNA by a eukaryotic cell or a prokaryotic cell into which it has been introduced should have the same amino acid sequence because the genetic code is almost universal and the same codon chart can be used for both prokaryotic and eukaryotic organisms. All right, I hope you know you didn't have enough time because that's clearly not enough time to answer an FRQ of that size. So go back to it after the video or when you have time to work on it. All right, so this is what we were, see, why do I have this here? Okay, maybe to explain what was just going on? Yes. Okay, so there is that codon 5, there's CGG, and so there we have CGG on our genetic code, and then we have arginine here, and that's why that transfer RNA. So CGG and the complementary anticodon is GCC. Now, the first thing that half of my kids want to do is look for GCC on this genetic code, and then they get the wrong amino acid. So make sure you're looking for the codon for the messenger RNA when you're figuring out what amino acids you should be bringing in. And then at 510, we have a UAG, and that is complementary to AUC. And if you look for UAG, I have it in aqua here, you'll see that that's where that stop is. Okay? All right, short debrief and summary today. I know I have went through a lot of information. Really didn't have that much information about, that would have been a really quick video about the pepper moth peppered moths and the sickle cell. So I added some things that could help you review along the way from some stuff you had at the beginning of the year and maybe in the middle of the year also. Anyway, so the takeaways for today: phenotypic variations significantly increase or decrease fitness, and that's reproductive success of the organism in particular environments. And you should be able to describe the importance of phenotypic variation in a population. You should also be able to explain biological concepts and processes. The common error, misunderstanding that could come from the natural selection phenotype variation part of this today is when you use the word fitness. If you're writing about this and you just write the word fitness, you're not going to score a point. You have to, in your words, show that you are understanding what fitness means, and that you need to talk about reproductive success and that there's offspring, and use those kind of vocabulary words to define what you're talking about. You should define a lot of things that you're talking about when you're writing, if you think there's any question about whether the reader will understand that you understand, okay? All right, so you're going to make reference to offspring inheriting genes for particular traits. And remember, don't speak Lamarckian. Now, you do have homework, all right? It's short, very short. Now, it's a one task verb. Certain human genetic conditions, such as sickle cell anemia, result from single base pair mutations in DNA. And you're going to explain how a single base pair mutant in DNA can alter the structure and some cases that function of a protein. So now we're talking about the sickle cell anemia. That was the trait, the phenotypical variation that we started this off with, and we moved this towards single base pair mutations and wrapped all of this biology together. So that's your homework for tonight. And I have 2 more slides I need to share with you. And when this gets saved, then just go back into it, and then you'll have that homework right at the very end, okay? All right, so this is new from the College Board, device and internet access. We know that not all students have access to the internet or a device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly to let us know. And here is the address, cb.org/tech. And thanks for watching. We hope you'll join us again. Be sure to complete your practice question. Ms. Kate will be tomorrow night with 7.3, and that's artificial selection. And we'll review the answer to the homework question. She'll start with that tomorrow in the next lesson. Okay, thank you.